Hello friends, welcome back to my YouTube channel Journey with Vijay Kumar Srivastav. Today we are going to study the topic agronomic principles in hybrid seed production. So let's move. The genuineness of variety is one of the most important characteristics of good quality seed. In scientific seed production, careful attention is paid at every stage to maintain genuineness and best quality of the variety. In spite of this, if not maintained, the varieties may become impure. It is therefore desirable to carry out determination for varietal purity to the extent possible. Examinations for determining genuineness of variety can be conveniently divided into the three groups. First one is laboratory examinations, second is greenhouse or growth chamber examinations, and third, field plot tests and field inspections, about which we will discuss in detail. The varietal purity tests which are carried out in a seed testing laboratory is called a laboratory examination. The test is based on the appearance of seed, means morphological characters. After that, seedling observations or chemical characters. The working sample consists of a total number of 400 seeds, where the 4 applications of 100 seeds for each are maintained and taken at random from a subsample drawn in accordance with the procedures defined. The morphological characters are examined with the aid of a suitable magnification. The color characteristics are examined under full daylight or light of the limited spectrums like ultraviolet light, and chemical characteristics are examined after treating the seed with appropriate reagent, and the reaction of each seed is noted. Now we will go through examination of seeds. There are different methods about which we will study here. First one is examination of morphological characters. The morphological characters of the seed grain can be observed by direct visual examination or with suitable magnification. The most useful characters in general are used as size and shape of grain or seed, base of lemma, ventral crease hairs, rachilla hairs, deviations of lateral dorsal nerves, wrinkling of lemma and palea, shapes and hairiness of lodicules, seed coat bloom. After that, hilum color in dicots and seed colors, which are useful characters that can be observed by direct visual examination under daylight or avid light. There are other methods like scanning electron microscopy (SEM), which are used for studying differences in seed coat surface and its inner structure, which have also to be used in some species. Now second method is chemical test. There are different tests which are used, like first one is phenol color reaction, second is modified phenol color reaction, third is seed coat peroxidase test for soybean crop, fourth is KOH bleach test for sorghum, fifth is ferrous sulfate color reactions in paddy, sixth is potassium dichromate or sodium hydroxide test for pea crop, and seventh is alkaloid test for lupinous species. And like that, there are different tests also which are specified for specific crops, and we will discuss in detail here. Now we will discuss about chemical tests which are carried out for determination of genuineness of varieties. Chemical tests have been successfully used in varietal identification of many crop species, and the major advantages of chemical test can be defined as it can be done easily by a technicians on a large scale in a seed testing laboratory in the shortest possible time frame, and these are relatively less expensive, and the tests permits detection of percentage admixture of other types. There are different methods of chemical test. First one is phenol color reaction. Phenol color reaction has been extensively used for varietal identification of wheat varieties. The advantage of phenol test is that it serves as an easy, quick and reliable test. And the procedure consists of soaking of seeds in water for defined period and as per the specific procedure defined, and the varieties could be grouped into different color reaction groups, like first one is nil or no reaction group, Second may have reaction as light brown. Third may be brown. Fourth may be category of dark brown, and it may be black reaction also. The admixture of varieties belonging to other phenol color reaction groups and vice versa could be easily determined. Second test is modified phenol color reaction, where the efficacy of phenol color reaction test could be further enhanced by the use of traces of copper and sodium, and the procedure is the same like phenol color reaction, but here instead of soaking of seeds in water, the seeds are soaked in low concentration solution of copper sulfate or sodium carbonate or any other chemical which are standardized for the test. Phenol color reactions has been used in purity determination in a number of crops like wheat, barley, oats, ryegrass, Kentucky bluegrass, and paddy, etc. Third test is seed coat peroxidase test for soybean crop. The seed coat peroxidase activity in soybean is controlled by a major gene. It is high when the dominant allele is present and low when the homozygous recessive allele is present. Since the individual seed is examined in this process, the admixture of seeds of high activity varieties into low activity varieties and vice versa could be accurately determined. Fourth test is potassium hydroxide bleach test for sorghum crop. The presence or absence of tannic acid, which is a dark pigment in the testa or undercoat layer of sorghum seed, which is determined by soaking sorghum seeds in solution of potassium hydroxide and fresh household bleach of sodium oxychloride for defined period, and the admixture of one group of varieties in the other could be determined. Fifth test is ferrous sulfate color reaction test, which are used in paddy. The paddy varieties, when immersed in the 1.5% solution of ferrous sulfate for 4 hours, which developed distinct color reaction spots, namely brown spots, grey spots, or grey streaks on the husk. The admixture of one group of varieties in the other thus could be determined. Sixth test is potassium dichromate or sodium hydroxide test for pea crop. In uncertain cases, the differentiation in the color sheets of garden pea and fodder pea can be made clearer by the using potassium dichromate solution or by a sodium hydroxide solution treatment. Seventh test is alkali test for lupinous species. In lupinous species, the presence or absence of alkaloids is a diagnostic feature. A distinct brown-red precipitate indicates the presence of alkaloids, and on that basis the determination is done. Biochemical tests are also used for determination of genuineness of varieties. First one is electrophoresis for proteins and enzymes. The electrophoresis terms refers to the migration of a charged particle under the influence of an electric field, where the movement of ions takes place in a suitable medium. The electrophoresis in a suitable medium separates the mixtures of proteins which are extracted from seeds, seedlings, or mature leaves into distinct bands. Each variety or genotype thus has a specific bending pattern on the basis of which admixtures of other varieties differing in bending pattern could be detected. The electrophoresis is now being increasingly used for determining the genetic purity of seed samples. In electrophoresis, the separation into distinct bands is based on differences in the size of molecular weight of various proteins. Particles with a smaller molecular weights migrate faster than those with higher weights. And second, on differences in charging. The molecules with the higher charge migrate faster than those with a lower charge. Second electrophoresis test is polyacrylamide gel electrophoresis, which is also called page. The degree of separation of the proteins isozymes through page varies according to pH conditions, concentration of acrylamides, and this ionic strength, potential gradient, and strength of the electric fields when the current and volts are applied. And the optimum conditions may vary for different species, hybrid varieties, depending upon the molecular weight of the proteins and isozymes which are involved, and hence needs to be standardized as per the crop and hybrid specific. SDS polyacrylamide gel electrophoresis test is also done for determination of genuineness of varieties. In SDS page method, separation of proteins are done on the basis of their molecular mass, which is done by denaturing proteins in the presence of sodium dodecyl sulfate SDS, which is a strong anionic detergent with the combination of thiologent mercaptoethanol, and then subjecting it to polyacrylamide gel electrophoresis test. Other chromatography tests are also done. First one is chromatography of flavonoids and phenols, where the variety differences based on the chromatography of flavonoids and phenols extracted from mature leaves, seedlings, and seeds have been reported. Extracted compounds are separated due to characteristics of solubility proteins by their different rates of migration in a solvent system. Flavonoids and phenols spot patterns on chromatographies are detected with the aid of several chemical treatments and UV lights. Other chromatography test is chromatography of fatty acids, and quantitative chemical assays have been also utilized as an aid in cultivar differentiation in several species. However, results are primarily quantitative and in some cases have been shown to vary due to environmental influences. Other test is ELISA test, which is known as enzyme linked immune sorbent assay. This is used for Bt protein test in transgenic crops to verify the genuineness of varieties or hybrids. Next test is molecular test, which is done to analyze the genetic purity, which is a check for certain genes, proteins, or other molecules which decides the genuineness of varieties. Now to study about examination of seedlings. The working samples are taken for 400 seeds, where the 4 applications of 100 seed each are maintained. In a determination process, the seeds are germinated in suitable media, and when the seedlings have reached in suitable stage of development, they are examined in whole or in part, with or without further treatment. For determination of ploidy, root tip is excised and processed for examination of chromosome numbers, and the method of examination of seedlings may vary for cereals, beet, brassica species, grasses, and other crops. Here we will study about examination of plants in glass house or growth chamber. In this examination, seeds sufficient to give not less than 100 plants are sown in suitable containers and maintained in the environmental conditions which are necessary for the development of diagnostic characteristics. So here temperature, moisture, aeration, humidity, everything is maintained as per the crop requirement. When the plants have reached a suitable stage of development, the critical characters are observed on each plant and noted. Sometimes the number of plants grown for observation will vary depending upon the space availability and crop growth. There are some advantages of this method, as the crops are grown in controlled conditions. In certain cases, it helps in observing more distinct characters and allows work to proceed almost all the year round. And in principle, this method doesn't much more differ from field plot method and takes less time as compared to field plot method. And there are some disadvantages also, like these are very expensive and it can be carried out on very limited number of plants. Examination of plants in field plots, where the grow out test is done for cultivar purity. The primary objective of grow out test is to determine the genuineness true to type of variety where plants are grown till observation recording phase. The determination is based on the observations of such plant characteristics of a variety which are least influenced by environment and are highly heritable. Usually, the variety differences are more distinct if growth conditions are favorable and plants have optimum ideal growth. In a field plot test, the conditions must be arranged in such a way that the genetical differences to be examined to demonstrate themselves as clearly as possible. Genetically, the observations are made on morphological characteristics, reproductive floral characteristics, and yield attributing characteristics. So, at suitable stage of development, the critical characters are observed on each plant. So, this is the method which is most commonly used for determining the genetic purity or two types of variety. There are some advantages and disadvantages of examination of plants in field plots. If we will discuss about advantages, it is the cheapest method to examine the reasonable number of plants, and it is possible to examine a large number of plots, and for each plot it is possible to check large number of plants. The plants are examined during the whole period of growth. Some characters are more prominent at one time of the year than another, and the samples may therefore be examined several times during the season. If you will discuss about the constraints and disadvantages, it takes longer time and the results are generally available after 3 months of sowing depending on characters to be observed and nature of variety growth period. And the crop is to be maintained well for each stage observations, and adverse climatic conditions may impact growth and clear data recordings also. Now the presentation ends here about determination of genuineness or true to type testing of varieties. Thank you very much.